Hi, thanks for watching and thanks for having me. I'm Mark, one of the co-founders and the Chief Commercial Officer at YURI, and I welcome you today in our beautiful YURI office here in the south of Germany. If it's ever allowed again, and if you ever happen to be in Germany, feel free to pass by anytime for a beer or for a coffee. We're happy to have you here. I'm happy to talk to you today about microgravity research. What are the benefits? What are the problems, the barriers we have today? Why isn't everyone doing research there yet? What are the innovations that companies like us bring into the process to make it cheaper, to make it quicker? And I'd also like to show you a few cool examples that we have that we're currently working on and payloads that we're going to launch. So let's jump right in. I'll promise you this won't be a boring presentation with tons of slides I'll show you. I'll try to make it more refreshing by showing you stuff here, by putting in video clips. So I hope you enjoy it. I hope you learn something from it. And I hope this inspires you to also think about your own space experiment, to think about your research or how it could help your research. So let's start with the benefits of microgravity research and how this might affect also your current running projects. And I'd like to give you two examples. As you know, microgravity is the state or the environment where you almost have no gravity. That could be on the International Space Station, ISS, which is the most prominent example, but it could also be on suborbital rockets or on parabolic flights. And without the parameter gravity, we have some effects, especially in the life sciences, that we can use. And one of those is looking at cell cultures. If you grow cells in gravity on Earth, then they normally just grow quite flat on a petri dish. And it's not really like tissue in a human body or in any body. But if you leave out gravity, if you grow those cells in microgravity, you basically have 3D cell cultures. You have cell cultures that grow in all dimensions because they're not limited by gravity. And so you have a more realistic picture, a more realistic model of how tissue works in the human body. And that is, of course, quite interesting in cancer research. If you look at tumor spheroids, that is quite interesting if you look at organoids and all types of tissue engineering questions. The other part is crystallization. So if you are a pharma company looking for a new drug, you look at the protein, the structure of the protein that is responsible for that disease. And because you cannot directly look at that protein because it's so tiny and a microscope wouldn't help you, what scientists do is you grow that protein in a crystal structure. That means you grow a large amount of that protein in a crystal, and then through a process called X-ray diffraction, you can determine the structure of that protein. Now, growing the crystal in gravity on Earth, you always have certain defects in that crystal structure because of convection and because of sedimentation. And therefore, you don't have a great, perfect crystal. And therefore, in the end, you don't have a very high resolution, a very high quality image of your protein. Now, if you grow that crystal in microgravity, you don't have effects like convection, sedimentation. So you can grow a higher quality with less defects crystal and also a larger crystal. And in the end, you can get a better image of your protein. And of course, that applies to all crystallization problems. So if you want to also grow fiber optic cable, which is basically also just a crystal, you can do that in higher quality, also microgravity, and anywhere also in the material sciences where you need to grow higher order, high quality crystals. So those were just two examples. So you can see there's many more benefits. But if there's so many benefits, why isn't many more researchers doing experiments and doing part of the research in the microgravity environment? Until today, it's still extremely complex, extremely time consuming to get anything to the International Space Station, to the ISS, to microgravity in general. And to understand this, I'd like to run you through quickly through the process of a typical microgravity experiment. And it all starts with the very first step, obviously, by defining the experiment you want to do, by defining what is the research question you want to solve. And there, typically today, you have lots and lots of paperwork, writing requirements documents. You need to write how many days, minutes, hours of microgravity you need, what temperature do you need, what volume do you need, and all types of requirements in very detail. And then only in the end, often you get a response from the space agency or from some other company that this is possible or this is not possible, and this costs X amount of money. The second step then, once you have done that, then you need to go to any hardware development company that can develop a so-called mini bioreactor for your experiment. And those bioreactors, they can run your experiment. They maybe have a culture chamber, a tank, a pump to run your experiment. And those bioreactors are typically, in most cases, developed from scratch for each and single experiment. And that, of course, makes them so expensive, because not only every new development is, of course, expensive. It costs a lot of engineering hours and material, but especially in space, because you need to qualify each and every screw and part and material by NASA, because, of course, they need to check if everything is, from a safety perspective, fine to fly. And that can take months or even more than a year just for this process to qualify and build your bioreactor hardware. Once you've done this, you move on to training and logistics. That means you run your experiment several times on Earth as if it would be in space to really train yourself how to grow yourself in the bioreactor. Does everything work? Because once you are at the launch site, there's not much you can do because the rocket doesn't wait for you. So you really have to have everything together once you are there. And then also, of course, there's lots of logistics now to clarify. How do you transport your biological material? How do you get stuff into a rocket? Which parties do you talk to? So there's a lot of management overhead there. Once you've done that and it's really gone through the process, the highlight of the mission is, of course, going to the launch site, to Cape Canaveral in most cases in Florida, cultivating your biological samples like cell cultures, for example, and then launching it on a rocket to the International Space Station. There, it's taken out from the crew. Some experiments have some crew interaction by the astronauts, and then typically you will receive your samples back after 30 to 60 days, and you can analyze them. Now, you already see this process sounds very long and sounds very complex, and it is. And typically you can calculate in terms of costs around $500,000 to $1 million per experiment for this whole process. And it usually takes at least 1.5 to 5 years to run this one experiment. And that is, of course, a reason why not so many people, not so many researchers have done it so far. And besides the big space agencies in normal, let's say, university research, institutional research, it's not super common yet. And this is exactly what we saw when we worked at Airbus a few years. So our YURI founder team, we've launched several payloads to the ISS. We've gone through this whole process several times. And we also saw possibilities, how to improve it, how to streamline it, and how to bring down those costs and timeframes a lot. And we are happy that we now brought this $500,000 to $1 million and several years down to roughly less than 100,000 euros per experiment. And in best case, we can launch it within 6 months. So this is a large improvement already. And I'd like to give you 3 or 4 reasons how we managed to do that. Number one is that we, instead of letting you for months writing those requirements documents and not giving you any feedback, we're currently also in development of an online configuration tool where you can enter parameters such as your microgravity duration, the volume you need, the temperatures, the sensors you need. And then with this tool, you can in real time get feedback on price and time and can play around with the parameters so you can really know what volume, what time, how much cost is needed for your experiment, and you can adapt your experiment to the cost and the time frame, which is quite convenient. The second part, and probably the most important part, is that we don't develop new bioreactors for every single experiment, but we build up a portfolio of modular and especially reusable bioreactors that are flight proven, that are already qualified, and that we can basically just refurbish, take it off the shelf, and fly it again. And that, of course, brings down costs a lot. And I'll show you one I just have lying beside me here. This is the YURI cube. It's a typical 10 centimeter cube, a 1U format, like a CubeSat. And inside you can see we have up to 3 bioreactors that we have in our portfolio. I can take this one out so you can see this is our typical form factor. And if I open this one, you see this is one of the typical bioreactors we fly. This is called the Type 4. It has a culture chamber here. It has 2 tanks and a pump, and you can run your cell cultures there. They can have refreshing media. They can have fixative media. You can have adherent. You can have suspension cells, and it's fully autonomous and it's flight proven. So every material, every part is qualified. So if you want to fly on this hardware, we could bring you to the ISS in best case in 4 to 6 months. That's one example, but we have many more of those. We have inserts or such bioreactors for fruit flies, for zebrafish, for crystallization, and for all types of experiments. And we're constantly developing more. So you can, in the end, and today already, just choose from a catalog of hardware and do not have to wait until new development is done, and you don't have to pay this new development. That is the big advantage. So the second thing we do is that, or the third thing we do is that we take care that if you just need a few minutes or a few seconds of microgravity, we do not necessarily have to bring you to the ISS, but we can also bring you on a suborbital rocket, which is a rocket flying up to space and down, which gives you a few minutes of microgravity. It's less complex from a safety perspective, and it's less expensive. And if that's enough for you, that is, of course, something we also do. And also, if you just need a few seconds and you might even join the experiment because you have to do some human experiment, we can even bring you on a parabolic flight. So an airplane that is flying on a parabolic arch and having about 20 to 30 seconds of microgravity. And that is, again, less complex and cheaper than an ISS mission. And if you just need a few seconds, this is definitely a better option for you. We also have a microgravity simulation machine called Kleinostat, and that's maybe also an important point. We take care of the whole end-to-end process. That means from start to finish, we take care of everything. You just focus on your science, and we take care of all the logistics, all the export control questions, all the communication with NASA and the launch providers. And basically, we are your lab in space, and you just work on the stuff that you need, and we take care of all the space topics. All right, so if that's interesting to you, of course, let us know anytime. And now I just, to end this presentation, want to give you a few examples on exciting missions we're currently working on. One of them is a payload we just launched last month in October, launched a payload to the ISS on a Northrop Grumman rocket, which was looking at protein aggregation to improve the shelf life of drugs. Another one will launch just next month in December on a SpaceX rocket, which is a project with NASA and with the UCLA in California, where they study neural stem cells and their behavior in microgravity. And the third one I'd like to mention here, also very exciting, is from an Australian researcher, from Joshua Cho from the University of Technology in Sydney. And he will look at how cancer cells, different types of cancer cells, behave in microgravity, and hopefully find some answers on how to better handle and even fight cancer with this research, with the answers he would get in microgravity. So this is what's happening on our side, and that's, of course, a great window of opportunity. So if this somehow can help your research, your developments you currently work on, we're super happy to talk to you. Let us know. We're happy to have a chat and exchange on current scientific projects. And if there's any questions, let us know on our website to check out more what we're doing, and if you want to get in contact. Thank you very much. Stay safe. Have a great conference this week, and all the best. Thank you, and regards from Germany.